Welcome to Virology. I am Vincent Racaniello. My last name rhymes with black and yellow. You know that song? One of the students in this course years ago said, that's how we remember it. It's perfect, right? Everybody gets it. Black and yellow, Racaniello. This is the 17th year I'm teaching this course. Oh my gosh. I love viruses very much. I think they're the coolest thing on earth, and that's why I teach this course. I'm a professor at the medical center, and I had a lab for 40 years that studied viruses. And I decided that there was no virology course down here. Is it ridiculous? All the money you pay to go to school here, there's no virology course. So I decided to start teaching it just on my own accord. It coincided with publishing a virology textbook. And so here we are today, many years later. I closed my lab, and now I'm focusing on science communication. So I'm going to teach this course to you, the whole thing. I do it myself, nobody else. I have two TAs. We have Irene, who's in the back there, and Claire is not yet in the country. She'll be here. But they will help you study for exams and so forth. My goal here is to teach you virology, okay? I don't want to give you a hard time. I want you to learn this because I think it's really important. I think this is one of the most important things you can learn in life, because as you saw, a virus almost took the earth down 5 years ago. So I want you to learn, so I do everything I can to help you. Right, the exams are all open book. You can take them wherever you want. All these things that you can drop one of the 4 exams. I do as much as I can. So here's the textbook. I'm one of the 5 co-authors, so I think this is a really good book, but you don't have to buy it to pass this course. You don't. It's just recommended. If you really are into it, you can find it used somewhere for a lot less than the price. But the course is based on this book. So what do I mean by that? The book, every chapter is a different principle. It's not a virus. You can't learn virology by teaching you different viruses, like flu and polio and herpes. No, that doesn't work. You have to learn principles, and that's what we have here. There are 2 volumes. We have like structure, genomes, infectious cycle, all the different steps in virus replication. And then we have issues about disease, adaptive immunity, vaccines, antivirals, evolution, and so forth. So it is based on principles, not viruses. So you learn a big picture about how viruses work. So one of the things that I have done since I closed my lab is to do a lot of science podcasting. And there are 2 related to this course. This is This Week in Virology, and there are 2 episodes a week. There's a basic science one, and you can tell here, Holy Cow, Convergent Evolution is the basic science one, where a bunch of virologists sit around and discuss papers. And then there's a clinical one with Dr. Daniel Griffin, who's an infectious disease physician, and we talk about what's going on with all kinds of infection every week. If you like to hear even more about viruses, actually, if you need something to fall asleep to, this would be good. A lot of people tell me they play it at night and they fall asleep to my voice, so you should check that out as well. So these podcasts are all recorded at a studio we have downtown. A number of years ago, I founded a company called Microbe TV. It's a nonprofit whose goal is to educate people about science. And now it's even more important than ever, right? When all the appointees seem to be anti-science people, it's really, really bad. And so one of our jobs now is to give people truth in science. So we have a recording studio that's where I spend most of my days. And we have all these podcasts, This Week in Virology, Parasitism, Microbiology, et cetera, that cover all aspects of science. And each one has a number of different experts in the field that always are there and talk about different science, basically. We have lots of listeners, both scientists and non-scientists, so this is filling a niche that we really need filled. We live and prosper in a world and a cloud of viruses. This room is full of them, you know. Gosh, you're all packed in here. You are exhaling viruses. I would wear a mask if I weren't 6 feet away. You're kind of dicey here, I think, but it's okay. I wear masks on subways now in the winter because it's the respiratory virus season. And for various reasons, I can't get a respiratory virus infection. I don't want to do that. But viruses are everywhere. They're in the air, they're in water. When you flush the toilet, you make an aerosol that is full of viruses. Yeah, poop viruses. And by the way, you should close the lid, but it doesn't help. The aerosol squirts out through the side and it fills the room. They did experiments where they put bacteriophages into the toilet water and flushed, and they could find it all across the room, even with the lid closed. Viruses are everywhere. They're in all of us. You have tons of viruses within you, yet most of you are fine. It's in our food. It's in every animal that you look at. And they're often part of genetic material, not just of us, but of different animals. So for example, and the numbers are the key to viruses, right? A virus infection is pretty a risky thing, right? The virus floating around trying to find a cell, that's that's high risk. So what they do is they make a lot of particles and hope that some of them find cells to infect. And so here's an example. In the waters of the world, mostly the oceans, but other waters as well, non-salt water, there are 10 to the 30th bacteriophage particles. These are viruses that infect bacteria. 10 to the 30th. That's like 1 million per teaspoon. So you go swimming and you like to take water in and spit it out, right? Because it's salt water and it feels cool. Full of viruses. It's not a problem for you because they're mostly viruses of prokaryotes and single-celled eukaryotes in the ocean, so they're not going to infect you. But there are lots of them. So we can figure out the biomass we know how much each one weighs, and we know how many there are, 10 to the 30th. It exceeds the biomass of elephants by 1,000-fold. All these viruses that you can't see exceeds the biomass of elephants. And then if you line them up head to tail, these phages, they will go 100 million light years into space. 100 million, that's past Pluto, the planet formerly called Pluto, right? It's past it. The nearest galaxy is 75 million light years. And these are invisible viruses. It's amazing. That just shows you how big 10 to the 30th is, because it's really hard to figure out 10 to the 30th. And by the way, that picture is what some bacteriophages look like. So numbers are the key to virus infections. Whales, for example, are commonly infected with caliciviruses. This is a family of viruses related to norovirus. You might be hearing that there's a norovirus epidemic going around. This is gastroenteritis. It's not fun. It's vomiting and diarrhea caused by a virus. But whales get it too. And they excrete over 10 to the 13th virus particles a day into the oceans, and other animals and non-animals can pick it up as well. So just the numbers are incredible. Now, if you look at particle number versus biomass, you get a very interesting story. And by this slide, I want to show you that viruses are not just bad news. In fact, most viruses, as we'll see, are harmless to most organisms. If you look at biomass in the oceans, so you measure the mass of prokaryotes, which is yellow, protists, which is purple, and viruses, which are blue. You see that the prokaryotes are by far the most biomass, right? They weigh the most in the ocean. But by numbers, that's abundance, viruses win. 94% of the particle number. If you just count particles, so a bacteria is one particle, a virus is one particle. The viruses win. There are tons of them. So why are there so many there? There's a lot of life in the ocean. And we didn't used to think there was much life in the ocean. But not only are the fishes and the mammals infected, but there's microscopic life that is infected by these viruses. And the cool thing is that the viruses help turn over the elements in the ocean. So they'll kill an organism, and it'll break down into smaller bits, and that can be further metabolized. And they're really key for different cycles, like carbon cycles and sulfur cycles and so forth. And without them, without viruses, these cycles would have a lot of problems getting completed. And we're not going to talk much about that in this course, but they are really essential for the biology of the oceans. Another number that's interesting, there are 10 to the 16th HIV genomes on the planet, human immunodeficiency virus, right? That's a pandemic that is ongoing still, because in many places it is uncontrolled. 10 to the 16th genomes, because we know how many people there are infected, we know how many genomes they have. So that's a lot of genomes as well. And what does it mean? It means that we already have resistance to all the antivirals we have. We have more antivirals against HIV than for any other virus or bacteria or antimicrobial. Yet, in these 10 to the 16th, we already have resistance. And if we keep making antivirals, we have resistance to all of them. That's how big that number is. So we're all infected to some extent. And we have at least a dozen herpes viruses in us. We're going to be talking about these in some detail. You get these when you are very young. And you all get them. Over 90% of the world's population is seropositive, which means you have antibodies for the virus, which means you've been infected. Here we have herpes simplex 1 and 2, varicella zoster, human cytomegalovirus, Epstein-Barr virus, and then other human herpes viruses. But all the other herpes viruses you typically obtain from your parents early on. They're shed in saliva. When they kiss you, they give you viruses. And they are with you for life. They establish what we call a latent infection, and you can't get rid of it. There's no way that we know right now to get rid of those infections. And we'll talk about the consequences of that. So you have all these herpes viruses in you, and you have, well, we all know we have microbiomes, right? Different parts of our bodies have different colonies of bacteria. Well, it's the same for viruses. It's just not been as well studied. So on the left, yes, the left, you can see a breakdown of all the parts of humans according to the viruses that are present. So the DNA viruses are in blue and the RNA viruses are in green. And we haven't talked about this yet, but you know, all life on Earth is DNA based. But viruses can be DNA or RNA based, and we'll talk about that later. So you can see in all parts of the body we can find viruses: nervous system, respiratory tract, urogenital, blood, digestive, skin, hair. Even your nails have viruses on them. So people have done these surveys and you can find them. So every part of your body is infected. And probably these are beneficial, but we just don't know it. We haven't been able to figure it out yet. Now, everything on the planet has a virome of some kind, and we're really interested to know what it is so we can understand its function. It's easy to know the virome of a bat or a bird or a mouse. You can just capture them. But what about whales? So here on the right is an experiment. You can see this is off the southern coast of Australia. And there are two whales here that have come to the surface, and they're breathing. And this is a drone right here with a petri dish on it. It's going to fly over the whales and capture their breath as they exhale. So let's see. Okay, it opens up. That whale just blew, and the petri dish is all wet. So now you can take whale breath back to the laboratory and figure out what viruses are in them. And it works. They found all kinds of viruses in whale breath. Now, we don't know. It's not representative of the whole whale, obviously, because it's just the respiratory tract. But you could never sample this. You could get a dead whale that washes up on the beach, but that wouldn't really tell you what's in a living whale. So we can do this. This kind of technology has really revolutionized virology. I mentioned earlier that there are viruses in your DNA. In fact, every animal on the planet, as far as we know, has viruses integrated into its DNA. And here's an example of the human genome. So it's a pie chart of our 3.2 billion bases of DNA. And the protein coding genes are very small, as you can see. They're the smallest portion, 1.5%. And then we have viral sequences. So these LTR retrotransposons, 8%. These are remnants of old virus infections. The genomes have integrated into the cell, and they're there forever. And as you'll see, sometimes we use some of these genes. Viruses, of course, do cause disease. So this is a chart of causes of death in the world in 2021. And you have to take this with a grain of salt because most countries don't have good reporting systems for health issues, right? only certain nations do. So it's biased. Yet you can see the top killer, cardiovascular diseases, then cancers, and then there's COVID-19. It wasn't there when I started this course, right? That's a brand new one. It may have been a respiratory infection. So you can see lower respiratory infections here, 2.18 million. That has a viral component for sure. And many of these may have components that we don't know about. Dementias most likely have a viral component. Here's HIV/AIDS, 718,000. So this is for 1 year only, and it's not up to date. And then down here, hepatitis and measles. Look at this, measles, 56,000 deaths a year. This is a vaccine-preventable illness. There should be no measles anywhere, right? We have a great measles vaccine. We just can't get it to everyone. And here we had a few hundred cases of measles last year because people choose not to vaccinate their kids. Go figure. That's going to get worse. Anyway, so viruses contribute to global diseases, and that's another reason why we're interested in them. But most of the viruses on the planet don't have any effect on our health or well-being. Now, why is that? Only a few viruses really make us sick. Well, why do the vast majority not? Well, there are a bunch of reasons. First of all, most viruses pass through us. The food we eat is full of viruses. Famously, cabbage has a lot of virus. So if you like coleslaw, you're going to eat a lot of insect virus DNA. So this insect here, the cabbage looper, it crawls over cabbage in the fields, and it excretes viruses that get on the cabbage. You just can't get rid of them. So when you make your coleslaw, you eat it, and it just passes through you. It's harmless because it's an insect virus. It cannot infect you. And then peppers have viruses, of course. All plants do, but peppers are apparently most popular because if you there's a company you can send your poop to, or your feces, excuse me. You can send your feces to a company, and they will tell you what's in it. They will sequence it. The most abundant RNA sequence in feces is a virus that infects peppers. It's called pepper mild mottle virus. It makes peppers look like this, so people don't buy them, but the virus is harmless. I get emails all the time from people. I had my feces analyzed, and there's this pepper virus. What do I do? There's nothing to do. It's harmless. It's just passing right through you. So a lot of the viruses will do that. Even viruses of meat that we eat will pass right through you. Now, some viruses are beneficial, and we have a couple of lines of evidence which shows that. Here's two situations which I really like. So on the left, we have this grass called Dicanthelium lanuginosum. So this is a grass that lives around hot springs. So you know, Yellowstone has all these hot springs, right? And there are grasses that grow around it. They're thermotolerant grasses. And you can show that the ability to grow at high temperatures is due to the fact that they're infected with a fungus, Curvularia protuberata, which in turn is infected with a virus. And you need both for the thermotolerance. So both the virus and the fungus are conferring thermotolerance to the plant. If you take them out, if you take the fungus and the virus out, the plants cannot grow at high temperatures, which here we're looking at 55C, they will not grow. So this is an interesting symbiotic mechanism whereby the fungus and the virus gets a place to live, and in turn the plant gets thermotolerance. Another example is shown here, which I think is very cool. This is a parasitoid wasp. These are wasps that inject their larvae into caterpillars. And the wasp has in its genome sequences encoding a virus. And when the wasp injects eggs into the caterpillar, so the eggs here are yellow, these ovoid yellow things, it also injects the virus, which are the blue particles. They're called polydna viruses. So these viruses do not harm the wasp. They do not harm the egg, but they infect the caterpillar and they immunosuppress it so that it doesn't reject the larvae of the wasp. and the larvae hatch, and they eat their way out of the caterpillar. This is what Alien was based on. Right? They didn't invent anything in that movie. This is real life Alien. They eat their way out, and of course the caterpillar dies, but now we have more parasitoid wasps. And the cool thing is that those viral genes are integrated into the wasp genome. So the wasp genome is weird. It's 2 circles of DNA, and the viral genes are integrated. So obviously this gave an evolutionary advantage to the wasp to have these viral genes in it. It's very cool. Okay, so that's great. Plants and wasps. What about mammals? How do viruses help mammals? It's hard to do, right? You can't do experiments on people. So you're left with animals, models, and this is one I'll show you, but we don't know the relevance to people. So this is an experiment with mice, actually done at NYU, where we take conventional mice and germ-free mice. So conventional mice are raised. You don't worry about what bacteria infects them. Germ-free, you don't have any bacteria in them. And we know that if you grow mice under germ-free conditions, their intestines are not developed properly. So here are the villi of the small intestine of a conventional mouse. They're nicely formed. And at the bottom here, we're staining for lymphocytes, and they're staining brown. And you see there are plenty of lymphocytes, as there should be in the villi. If you grow mice under germ-free conditions, you can see the villi are morphologically aberrant, and there are very few lymphocytes below. So the absence of bacteria causes the intestine not to develop properly, nor the immune system. Now, if you infect these mice with norovirus, murine norovirus, which is an RNA virus of mice, it partially restores these defects. You now have lymphocytes in the villi, and the villi are somewhat more normal. Not 100%, but looks, and again, these are germ-free mice that have just been given a virus. So it can partially restore the defect that happens if you don't have microbes. So this is kind of suggested that there should be beneficial viruses for us. It doesn't prove it, obviously. Someday we may have broad-spectrum antivirals like we have broad-spectrum antimicrobials. And then maybe we'll see if viruses are beneficial, because we'll wipe out all the viruses in us and we'll see what happens. Kind of like what happened when we developed broad-spectrum antimicrobials and we had problems arise. Now, the other reason most viruses are okay in us is because our immune system is really amazing. And this is just a little summary of the immune system. We will go into it just a bit in 2 lectures, but the immune system is great at keeping those virus infections in check. And if your immune system is down, if you get immunosuppressed, for example, and there are a bunch of viruses that cause immunosuppression, or if you're getting an organ transplant, you are immunosuppressed so you don't reject, many viruses start to multiply. All these herpes viruses start to multiply, and they can cause serious problems. There's a whole field now. As you know, transplantation is a big deal now. We know how to do it really well, not just for hearts, but for lungs and livers. And we immunosuppress people. So now there's a whole field of transplantation infectious diseases where people have to manage the infections that arise in transplant recipients. Now, along the theme of not all viruses make you sick, I wanted to show you this result, which is interesting. This is a virus called human polyomavirus. We will come back to that later and talk about what it can do. But this virus is passed among family members. And you can actually track the movement of people by seeing which variety of the polyomavirus they got, because there's certain different varieties that you can distinguish. You can actually track human migration by seeing what polyomaviruses people were infected with. So here's a map of human migration out of Africa. So the purple dotted line is the traditional map of human migration that we've used by looking at people themselves. And you can see we move out of Africa into Europe, across Asia and into the Americas. But the black lines are the migration of peoples based on what polyomavirus they have. And it's much more detailed. As you can see migration down into Australia and also into South America. And so these don't make you sick, but they infect you and they give us a marker for tracing population movements. Viruses also shape populations. Viruses shape populations and also vice versa. Many, many elderly people were wiped out by SARS-CoV-2. It had a big impact on the elderly. But in nature, viruses impact other organisms. This is a satellite photograph of eukaryotic phytoplankton. So these are microbial species, but they're eukaryotes that grow in the oceans, huge numbers, so that you can see them from space. And the white is the huge blooms of these phytoplankton. As soon as these blooms arise, which typically happens when there's lots of sunlight, there's a virus among them that will infect them, and the bloom collapses, which means it's disappearing. So the virus has this huge effect on these populations, and just an emphasis on what viruses can do. So those are just some examples of why I think viruses are amazing. And studying them is very interesting because virology is an integrative science. What does that mean? So you take chemistry, you learn chemistry, and that's it. But to understand viruses, you need to understand all the chemistries, biochemistry, organic chemistry, inorganic chemistry. You need to know molecular and cellular biology, physiology of organisms. You even need to understand sociology to understand how people interact and spread viruses among them. So it's an integrative science. I've had students tell me that all of the science courses they take now make sense in the context of virology. So you learn a lot more than just viruses because they are obligate pathogens of their host. So this course, what I want to do is give you a big picture of virology. I want to teach you about how viruses work. I'm not going to teach you about individual viruses for the most part. There are a few that we'll learn about, but I want to teach you how viruses work in general. And in the end, you're basically going to learn things that frighten most people. You know, when people get frightened about something, it's usually because they don't understand it. So you're going to come out of this class with an understanding of viruses. You're going to be able to know what's going on in the next pandemic a little better than most people. You know, there are many potential pandemic viruses out there. And, you know, a big problem is the press and how they present it to you. You're going to be able to filter through all of that after this course. So here, for example, are some headlines from newspapers. I have a big problem with the reporting about viruses in the press because most journalists are not scientists and they're not virologists, of course. So here at the top is the beginning of the pandemic. Coronavirus could mutate. So you will learn in this course that viruses mutate every second. Every replication cycle, they're mutating. So this is not the right headline, and they don't understand it. They have one behind it, below it. The coronavirus is mutating and America's leaders are flying blind. This makes no sense whatsoever. Then, on a different topic, gain of function. This has become politicized. Republicans spin NIH letter about coronavirus gain of function research. House approves ban on gain of function pathogen research. I don't think anybody in Congress knows what gain of function means. Right? Gain of function is simply giving an organism a new property. That's it. It could be a virus, a bacteria, a fungus. It could be a mouse. If you want to make better beer, you engineer the yeast and you give it a gain of function. They don't get this. They think it's all bad. In fact, there was the Congress released a couple of weeks ago a report on COVID, right? It's their interpretation of what happened. And it covers a lot of different things like forced masking, which is such a bad thing. That's sarcasm. And they said the number one reason why SARS-CoV-2 clearly came from a lab is because that lab was doing gain of function research. This is the dumbest statement I have ever heard. It's ridiculous. because gain of function can be so many things, and even if they were doing it, which they were, actually, it doesn't mean that that was the origin of the virus. In fact, it wasn't, but we'll talk about that later. Anyway, you're going to be able to sort through all of this, I hope, because why? I'm going to tell you information, not misinformation. You're going to learn that about viruses. Okay, in the interest of time, I will show the results. I think you can keep answering, but obviously once I show the results. So most of you got B. Our immune system can manage most viral infections. One person said the press is correct just to bother me, right? Yeah, that's fine. I don't have a problem with that. It happens every year. Anyway, we're going to do that throughout the lectures. So what is a virus? Here we've been talking a lot about viruses. What is it? An infectious, obligate, intracellular parasite. It's made up of DNA or RNA, and it's surrounded by a protein coat. And sometimes it has a membrane as well. So infectious, you understand what that means. It can go from organism to organism and enter an organism and reproduce. Obligate intracellular parasite means a virus has to get inside of a cell in order to make more viruses. Obligate intracellular parasite. And then they all have protein coats. So here are a variety of different viruses. And you have some viruses that just have a protein coat, like these. And then here's a coronavirus at the bottom. And the RNA is coated with protein, and then it has a membrane around it. So a lot of viruses have membranes as well. So that's the definition of a virus. And because they're obligate intracellular parasites, when you study viruses, you learn a lot about the cell, right? Because you're always studying it within a host, and a host can be many things. Now, are viruses alive? Here's the answer to that. It's a little complicated. A virus is an organism with two phases. So there's the virus particle here, or virion, the infectious particle. This is not living because it will sit in a tube forever and not do anything. It won't multiply on its own unless you put it in a cell. So the second phase of a virus is the infected cell, and that's certainly living. It's been reprogrammed by the virus to make more viruses. So a virus is an organism with two phases. A non-living phase, which is the virus particle, and the living phase, which is the infected cell. Okay, so that makes it very easy. I want to make sure that you don't treat viruses like people. Okay, don't use anthropomorphic analyses, because viruses are not people. You don't know what motivates them, right? You have no idea. So they don't think, they don't try to do things, they don't employ, exhibit, display. They're just passive agents that bump around and bump into a cell, get taken up, and things happen. There's no intent. So to say, oh, the virus mutated because it wanted to do this. Nope, don't say that. Because you have no idea what drove that event, because you're a human looking at a virus and you have no idea what's going on. So the reason I tell you this is if you apply human behavior to a virus, it's going to cloud your understanding of their evolution. So try not to do that. Now, viruses are small. Here's some illustrations to show that. Here's an E. coli at 100,000 X, and there is a bacteriophage that infects the E. coli. Here's HIV in D. And then here is a panel that is expanded to a millionfold here, and here in H is poliovirus, and there's a ribosome right above it, and there's a carbon atom in A. So you can see they're very small molecular size. Many millions of viruses can fit on the head of a pin. And so this calculation has been done. For rhinoviruses that give you common colds, you can put 500 million on the head of a pin. And there's the head of a pin right there. This is a dust mite, by the way, this orange thing. And then we zoom in here. There are all sorts of things here. Here are red blood cells and bacteria. But the viruses are right there. They're quite small. Now, about 20 years ago, we learned that not all viruses are really small. In fact, there are some giant viruses, and you can see them here. So here is HIV, and here is a rhinovirus. which causes common colds. And this Mimi virus was discovered in 2000, is that 13? 2011. You can see it was much, much bigger than these viruses here. So since then, we've found a lot of giant viruses, and they illustrate some new concepts in virology that we'll go through. For example, here are some of these giant viruses. Here is Pandora virus, so called because the genome was sequenced. It was like 2.5 million base pairs of double-stranded DNA encoding proteins that we'd never seen before. We have no idea what they do, so they called it Pandora virus. You can actually see these in a light microscope. They're about 1 micron in length. Most viruses you cannot see in a light microscope, but this is an exception. And there are lots of other giant viruses with very large genomes. Here's Mimi virus, and you can see the virion size increases with genome size increasing. Now, there's a key feature of viruses that is different from all other life forms, and that is they replicate by assembly of components. So bacteria in cells do binary fission, right? You take a bacterium, you put it in a culture, and very rapidly it begins to divide. The bacterium becomes 2 and 4 and 8 and 16. That's binary fission. Viruses don't do that. When a virus gets into a cell, it doesn't split into more viruses. But the virus gets in the cell. Its genome is used to make virus proteins. And those are assembled into new virus particles. And that's how viruses are made. And so as a consequence, in bacteria, if you put bacteria into a broth culture, they begin to divide right away. But if you infect cells with a virus, there's a period when you see nothing because the virus has entered the cell and it's beginning to make new virus particles. And then only after a certain time do you see the synthesis of new virus particles. So this is a fundamental difference between viruses and everything else. And we'll come back to this later and understand what it's doing. Okay, for Socrative number 2. Okay, which of the following is true concerning bacterial versus viral replication? Viruses must assemble using preformed components. Bacteria do not replicate via binary fission as viruses do. Bacteria must assemble using preformed components. Viruses do not have an eclipse period. Viruses replicate by binary fission. So I should show you what an eclipse period is. So this time when you infect cells with a virus and you don't see any new viruses, that's called the eclipse period. So viruses do not have an eclipse period. I already answered that for you. So, all right, let's see how we did here. So the answer is A, viruses must assemble using preformed components. Right? The virus infects the cell, makes components, and then assembles them. Bacteria do not replicate via binary fission. That's not true. They do. Bacteria must assemble using preformed components. No, that's what viruses do. Viruses do not have an eclipse period. I showed you that they do. Okay? They have an eclipse period. Whereas bacteria don't. You put them in broth and they pretty much start to divide right away. Viruses replicate by binary fission. They don't. They replicate by assembly of preformed components into viral particles. Okay, so how old are they? This is a tough question. So we will talk about how you determine how old a virus is later, but we think we can find signatures of viruses 450 million years ago. That's the Ordovician period. That's where we saw the first land plants arising. And these viruses probably infected organisms like this in the oceans. But the ancestors of viruses, the ancestors, probably originated before cells, which would be billions of years ago. And so we will have a lecture on evolution to explain that. But the idea is that there were self-replicating nucleic acids in the early Earth, which eventually became cells. Now we can see more recent reference to viral diseases, 700 BCE, 1500 BCE. That's a Greek vase on the left with a reference to rabies on it. And there on the right is a Egyptian carving with a priest who looks like he has polio in his leg, which the virus causes paralysis, and we'll talk more about that. So we can see references to viral diseases in recent history. Variolation is seen in the 11th century. So smallpox was a big problem globally for a long time. And in the 11th century, it was a big issue in China. What they learned was that people who already had smallpox and didn't die never got it again. So they thought, let's give people smallpox and see what happens. And the smallpox is manifested by a rash. So they would take the pustules, grind them up, and you can see they're blowing it into the nose of that individual. And it has virus in it. It would infect you and kill about 30% of the people. But the 70% would live. So it's kind of the first vaccine, but not with a good safety profile, right? 30% lethality. Lady Montagu, who was the wife of the British ambassador to Turkey, she learned about this. She brought it back to the UK. In the 1790s, Edward Jenner did an experiment to show that what you're really doing is vaccinating people against the virus. And we'll talk more about that when we talk about vaccines. The whole idea of microorganisms and viruses arose with the work of these three individuals. So Leeuwenhoek developed the first microscopes that could see in quite good detail. And so he said, there are things in the world beyond what you can see. Not everything is just what you see. Pasteur said, these tiny things are bacteria. We can grow them in culture, and they can do cool things like make wine and cheese. So he established that microorganisms exist. And then Koch, the last individual, said, these bacteria, they can do cool things, but they can also make you sick. And he established the principles of identifying how to say this bacteria causes disease. Now, where does virus come in? You can find in the literature as early as 1728 that virus is used to describe any agent that causes infectious disease. So in 1728, we don't really know about bacteria and viruses. We just know there are diseases that go from person to person, infectious diseases. And so these were called viruses, because virus is Latin for poison. And they were thought to be liquids that went from person to person somehow. And Pasteur, in fact, later said, he made it more refined. He said, every virus is a microbe. It's not necessarily a liquid, but he's thinking of bacteria, right? A key event in virus discovery is the development of this filter, a Chamberlain filter. So this is a guy who worked with Pasteur in France, and the water in the lab sucked. It was dirty and contaminated, so he designed this filter to sterilize it. It had a vacuum attachment, and this was a porcelain filter. You would put your water in the top, and it would remove all the bacteria. So this withheld bacteria. So Pasteur was working on rabies, and he put the rabies agent into this filter, and it went through it. So he said, it must be a small bacterium. You know, Pasteur was really good, but he just didn't get this part. He thought it was a small bacterium. Okay, so fast forward to the end of the 1800s, people are already smoking tobacco, and there's a disease ravaging tobacco called tobacco mosaic disease. And because it's economically important, a lot of scientists are working on it. And what they do is they grind up the leaf and try and put it through this filter. Well, two individuals, Ivanovsky and Bayerink, found that the agent of this tobacco mosaic disease went through the filter that retains bacteria. And they didn't just say it was a small bacterium, because what they also found was that this agent needed a host to grow in. It wouldn't grow in broth. You needed to put it on the leaf of the tobacco plant. So they got it. They said, this is something different. This is a filterable virus. So that's what they called it. So they use these Chamberlain filters, which have a pore size of about 0.2 microns. Today you can buy them in the lab, right? These are nitrocellulose filters. You can buy 0.2 or 0.4 microns. Same principle. So filterable viruses was the name given to these agents that pass through the filter. 1998, foot and mouth disease agent was identified. Foot and mouth disease was a disease of cattle, still is. That's economically important, so people wanted to know what it was. And again, it goes through the filter and it needs a host to replicate in. These were 0.2 micron filters, so these are very small agents going through. And the key is they still thought they were liquids, even though Pasteur said they were microbes. So here, filterable viruses, they're called for a long time. 1901, yellow fever, rabies, smallpox virus, influenza, not till 1933. So the pace of virus discovery spurred on by these filtration experiments. But, and here's the pace of virus discovery compared to other agents. Here is a timeline from 1830 to 1935. So here's virus discovery in red, and they're called filterable viruses, right? But the discovery of bacteria leads to an explosion in the identification of bacterial species. 1939, this is the key. So Helmut Ruska in Germany had built the first electron microscope, and he took pictures of viruses. He took pictures of viruses infecting bacteria. And he said, whoa, they're not liquids, they're particles. They're actual physical particles. So viruses are not liquids. We can discard filterable viruses from this definition. We just call them viruses from now on. And they're different from bacteria. And this experiment that I've already told you about also proved that viruses were not just small bacteria. This was done in 1939. You infect cells with viruses. There's an eclipse period. That's different from bacteria, where you put a bacterium in a culture and it immediately starts to divide because it's binary fission. The viruses have to make the parts and then assemble viruses. So they're fundamentally different. They're not small bacteria. So that's why we call them viruses today. And they're fundamentally different from bacteria. All right, our last questions for today. Which is a key concept first discovered about viruses that distinguish them from other microorganisms? Too large to pass through a 0.2 micron filter. They could replicate only in broth. They made tobacco plants sick. They were small enough to pass through a 0.2 micron filter. None of the above. All right, let's have a look. They were small enough to pass through a 0.2 micron filter. That was the key, initial key concept that distinguished them. Not too large. They don't replicate in broth, right? Viruses do not replicate in broth. You need a cell. They made tobacco plants sick. That's not really a distinguishing feature. They were small enough to pass through a 0.2 micron filter. All right. Bless you. So nowadays we know what viruses look like. We can take pictures of them in an electron microscope. There's an A bacteriophage, and B is tobacco mosaic virus. That's the virus causing the disease of plants that was eventually discovered. C is rabies virus, and D is a rotavirus. We know many details. We know the structures of many viruses. This is polioviruses at different resolutions of microscopy, and we will talk about how we can get pictures like this and what they can tell us. And we can classify viruses. We used to use many different properties to classify viruses, but today all we do is sequence the genome of the virus, whether it be RNA or DNA, and immediately we can classify the virus as either something that we know about or something that's totally new. In the old days, we used to talk about what kind of shell the virus had, whether it had membranes, the dimensions of the particle, but these are all gone now. The computers have taken over, and you put the sequence in a computer, and it says, this is a new influenza virus, or I've never seen this before. Make a new family to put it in. So for example, the way we classify an Ebola virus is shown here. The family, so we start at the family level. It's Filoviridae, or the filovirus family. And Ebola viruses are unusual. They're thread-like, hence the name philo. Then we assign a genus, like Ebola virus. And then sometimes we have a species, Zaire Ebola viruses. So the species concept is not really valid for viruses. Species are supposed to be things that cannot mate and produce offspring, but viruses don't really do that. So we just use species as a taxonomic level to say, here's an example of a Ebola virus. And you'll see examples of this throughout the course, so this is what we'll be talking about. Now, you can see from the slides I've shown you that disease motivated virus discovery. I showed you that picture of all the early virus discoveries: yellow fever, smallpox, polio, influenza virus. So whenever there was a disease, people said, what's causing it? And they found a virus. But we do that much less now. Yes, when there's a new pandemic, we say, what's the virus causing it? But mostly we want to know what's out there. So this virome, the virosphere, which is all the viruses on the planet, what's in it? And people just take samples and look for viruses. They take samples, they determine the genome sequence, they either look for RNA or DNA, and then they use computers to discover viruses. So this is a recent paper that we covered on one of our podcasts, using AI to document the hidden RNA virosphere. So they took metatranscriptomes. So they'll take ocean water or dirt or plants that other people have sequenced to find the RNA transcripts. And they look for a specific protein, an RNA-dependent RNA polymerase, which is unique to RNA viruses and is the most highly conserved protein. Now, previously, most people would take the sequence of this protein and use it to compare. But they trained an AI program to look at the structure of the RNA polymerase. And you can find RNA polymerases that way, which have no sequence identity, yet the structure is the same. It has an active site and all of the accessory structures that make it an RNA polymerase. We'll talk more about that later. But the point is, they were able to identify many new RNA viruses that hadn't been found before. 161,000 putative RNA virus species, which is huge. Now, this is an example of how AI is transforming virology. They say putative because they have just a sequence, right? They don't have virus, so they don't know if the virus actually exists. And that's something that we will come back to later, because just having a sequence doesn't prove that there is a virus around. Anyway, this figure is very cool because you can see the gray are the known viruses that they identified, right, in this figure. So there are not many of them, compared to the expanded virosphere, in other words, the new ones they discovered, all in blue. So they discovered way more than exists by this approach. And this is just for RNA viruses, so presumably it could be used for DNA viruses as well. So this is the future of virology, to understand what's out there, the relationships among them, how they evolved, and whether they have any potential to cause human disease. So why do we care about this? There are a lot of reasons, but these are what I think are the most impressive. Viruses outnumber cellular life by 10 to 1. I showed you that pie chart early on to support that. They have the greatest biodiversity on the planet. Nothing comes close to it. They drive biogeochemical cycles, as I mentioned earlier. They shape host populations and vice versa. And they're probably beneficial. And then, of course, they're sources of new pathogens. So we always have to be on the lookout for the next pandemic virus. It's not easy to do, but that's why people want to know what's out there, so we're not surprised. So this may seem complicated, but in the next several lectures, we're going to use two simple facts to simplify how we think about viruses. First, that all viruses are obligate molecular parasites. They have to get into a cell in order to reproduce. And they have to make messenger RNA that can be translated by the host cell. Viruses cannot make their own proteins. They have to use the cell's protein synthesis apparatus, so the mRNA has to be suitable for recognition by a ribosome. So we're going to use this to establish a framework of viruses in the next couple of lectures. And next time, Monday, we will talk about the infectious cycle.